Hello, my name is Alina Murphy. I'm a PhD candidate in the OBGYN department, and my goal is to determine how obesity leads to cancer. Obesity, which is defined by a body mass index of 30 or higher, is a growing problem in the United States and worldwide. Obesity predisposes people to many diseases, including cardiovascular disease and diabetes, but many people don't realize that obesity also predisposes people to develop cancer, including breast and colorectal cancer. The type of cancer that's most strongly associated with obesity is a type of gynecological cancer called endometrial cancer, and this is what I study. In the next few slides, I'll introduce endometrial cancer and talk about how I study it in the lab and how I hope to determine how obesity promotes it. Endometrial cancer develops in the uterus from the lining of the uterus, which is called the endometrium. And this lining grows and sheds each month as part of the menstrual cycle. When the cells grow out of control, however, a tumor can result. Endometrial cancer is the most common gynecological cancer, and it's very strongly associated with obesity. Obesity is now known to promote endometrial cancer. More than 50% of cases of endometrial cancer are thought to be caused by obesity. This graph shows how the risk of endometrial cancer increases with increasing body mass index, or BMI. BMI from 20 to 25 is considered healthy, and BMI over 30 is obese. You can see that the risk for both endometrial cancer incidence and mortality increases sharply in the obese range. Endometrial cancer incidence and obesity are both expected to increase, and so it's very important to understand how obesity leads to endometrial cancer. So why don't we know why obesity causes cancer yet? Well, one problem is that current cell culture techniques to grow endometrial cells in the lab are not very good. Usually, endometrial cells are grown in 2 dimensions on a flat plastic plate. Only one type of cell is grown at a time. But in the real endometrium, there are actually 2 main cell types which interact and talk back and forth to each other. It's important to study both types of cells to understand how the endometrium actually works. My lab has developed a new endometrial culture system in which we grow 2 types of endometrial cells in a 3-dimensional model that we call an organoid. So the one type of cells, represented by the pink cells, are located in the center of the organoid, and they're surrounded by the orange cells, the other type of cell. These endometrial organoids are a better model of the real tissue, and they are long-lived, which enables us to treat them and study how they behave over a period of several weeks. To create our endometrial organoids, I isolate healthy endometrial tissues and grow the cells from those tissues in 3 dimensions. These pictures represent a thin cross-section of healthy endometrial tissue and a thin cross-section of an endometrial organoid. The brown and purple spots represent the center of the cells, or the nuclei, and the pink spots represent one of the 2 main endometrial cell types. You can see that the structure of the organoid is very reminiscent of the structure of the actual tissue. I use these cells and organoids to determine how fat might change the function of the normal endometrium. So I grow endometrial cells in organoid form, either alone, or I grow organoids with adipose spheroids, or fat spheroids. I treat them with menstrual cycle hormones to replicate the menstrual cycle, and I ask, what changes does the fat produce in the healthy endometrium? And how might the endometrium now be predisposed to develop into cancer? My results are still preliminary, but they suggest that the presence of excess fat might be changing the way the endometrium responds to hormones. The endometrium is very sensitive to hormones, which control the way it grows. And in particular, the hormone progesterone plays an important role in stopping cell growth in the endometrium. So therefore, it's extremely protective against endometrial cancer. These pictures show an endometrial organoid which has been grown without fat, or an organoid that's grown in the presence of 30 fat spheroids. The brown color represents a protein that's produced when the endometrial cells encounter progesterone. You can see that there's less brown in the organoid that's grown with 30 fat, showing that the organoid is not responding to progesterone as well. So if the presence of excess fat is preventing the endometrium's protective response to progesterone, this could be one way that obesity is leading to endometrial cancer development. So, although we still don't know exactly why obesity leads to endometrial cancer development, we are making progress because we have developed a new, better system to model the endometrium in culture. We believe that this endometrial organoid model will be applicable in many contexts to study how the endometrium behaves in the context of obesity or cancer or other gynecological diseases, even testing drugs. We think that this will lead to improved therapies and better outcomes in the future. Thank you.